So thus the new molecule of DNA formed would be made up of one old strand and one new strand, and this method of formation of new DNA molecule is called as semi-conservative. Now please pay attention to this diagram. This is the original DNA because we already have understood what is duplex. So you can see it's a duplex because there are two strands of DNA running parallel to each other, anti-parallel to each other. So one will be 5' to 3', other will be 3' to 5' to 3', other would be 3' to 5'. So you can see here theoretically I have just mentioned here GAA, GTC. So if it is G on one strand, it should be C on the other one with three hydrogen bonds. I have only drawn one to make it clear. Then A will have opposite T, then T will have opposite A, and C will have opposite G. You have already learned these things in 11th and 12th. So now what exactly is semi-conservative? Imagine these two strands which are the old strand or the parent DNA molecules. These two strands, when they separate from each other, both can act like template. So this is the old strand and this is the old strand. We know the rule of complementarity. So if this is the old strand, then the new strand opposite to this will have C, T, T, C, A and G. And in the same manner, because this is the old strand, then the new strand will carry C, T, G, A and G. So now what exactly has happened? One molecule of DNA has now given rise to two molecules of DNA, but when you pay attention minutely, you will find both these molecules are exact copies of the parental strand. This has happened because of which property? Complementarity. So now you can see that in this molecule of DNA, one strand is old, that's why I have drawn it boldly, and the other one I have drawn thin. One strand is new, synthesized, and the other one is old. In this case also one strand is old and the other one is the newly synthesized. That means that if this is the original piece of DNA with two strands, now in this case one old strand is present here, the other old strand is present here, and these two are the new strand. That means that the new molecule formed would be made up of one old strand and one new strand, one old strand and one new strand. This method of formation of the new DNA molecule is called as semi-conservative. I hope this property is clear to you. This concept was proposed by Watson and Crick according to the NCERT also, but they only gave a theoretical proposition. Who confirmed it? There were scientists who performed experiment, and they proved by experiment that the DNA replication is semi-conservative, and those two scientists were Michelson and Stahel. So Michelson and Stahel in 1958 performed this experiment. This is explained in the NCERT also in the same manner. What they did, so their material used for experiment was Escherichia coli, which is found in the human bowel, comes out with the feces or excreta or waste material. So they grew E. coli for many generations in a medium containing nitrogen salt. So which salt was it? The ammonium chloride, but the nitrogen used is not the normal nitrogen, because normal nitrogen should have N14 mentioned here. This is N15 mentioned here. So it is a heavy isotope of nitrogen. Now what they did, they took E. coli, a normal E. coli, and now we all know E. coli is supposed to divide after every 20 minutes into 2 and then 4, it continues like this. So a complete generation or a population or a colony is formed. So what they did, they took one E. coli and they grew this E. coli in a medium which had everything normal except nitrogen. The nitrogen they provided to the E. coli was a heavy isotope of nitrogen, N15. So now what will happen? Every time E. coli would divide, it will use material from the surrounding, and then obviously whenever DNA is going to be synthesized during the synthesis phase of cell cycle, instead of N14, it will incorporate in its DNA N15. So slowly what will happen? A time will come till bacteria DNA will become completely labeled. So a time will come, a generation will come when all DNA in all these E. coli would be heavy DNA, or you can say all of them will carry N15 in it.